Hello everybody, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And what we were discussing about the topics from your biology course for the class 11 and 12 under the program IIT PAL. And in this series, we were discussing about the animal kingdom. So if you recall, in the previous lecture, we discussed about the how the animals are diversified, how they are different in terms of their habitats, their feeding habits, and their locations. And in that series, we also discussed because the animals are so much diversified, there is a criteria which you have to use to classify them. And we also discussed what is the advantage of classifying animal kingdom or the animals what is present in this particular kingdom. The major advantage of the classifying the different animals into the present in the animal kingdom is that it actually allows you to understand the relationship between the different animals. It also allows you to understand how the part of one type of animals are being evolved from the another animal, or how the animals are being connected to each other. And apart from that, when you actually classify the animals, you can be able to easily be able to understand their features and their other properties without even going through the much detail. For example, if you are identifying an animal which is actually belonging to some class of the particular animal kingdom or particular phylum, then you know that okay, if this particular animal is belonging to Porifera, it is actually going to have these are the characteristics it should have. And if some of the characteristics are present, then you are very much sure and you can be able to even extract more information because you have classified all other animals. So apart from that, because the animals are very diversified, you always have to use a very detailed and discrete criteria in terms of how you can be able to classify the animals. So what we have discussed that in the animal kingdom, which are actually multicellular, you can have the different types of criteria. For example, you can have the level of organizations, you can have the symmetry criteria, you can have the body cavity or coelom as a criteria, or you can have the even how the animals are, whether it is having the diploblastic or triploblastic kind of development. So in today's lecture, we are going to discuss some of these, animal classifications and and we are also going to understand about the characteristic of each phylum. So we start with a very simple thing. So we'll start with the cellular level of organization. So if you see there's a cellular level organization, you only have the one phylum which is Porifera, and that belongs. So you see everything is premature or primitive. For example, it is using a cellular level of organizations. The cells are asymmetrical and it does not have a coelom or the body cavity. And the classical example or the phylum which belongs to this particular property is the Porifera. So let's start discussing about the Porifera. Porifera are the aquatic animals, mostly the marine, so they are present in the sea and ocean, except that the family Spongilidae, which actually lives in the fresh water. They are so exception are there because these are actually either the sessile or the sedentary, and they grow like a plant. So these are the animals which does not follow the basic principle of the animal that they are should be motile. So these are the poriferas are the sedentary and they grow like a plant. So they get attached to any rock or something, and then they grow on these particular rocks, and that's how they are actually growing like a plant. They are multicellular organism with the cellular level of organization. So there is no organ or there is no tissue present in the porifera. They consist of the two germ layers. They are present as the outer ectoderm and the inner endoderm with an intermediate layer of mesenchyma. So they are typically having a diploblastic animals, which means they only have the two germ layers, and that's how they are belonging to a diploblastic layer. The contractile vacuoles are present in from some freshwater vacuoles. So you remember that the vacuoles are always been present in the plants. The sponges processes a high power of regeneration. So sponges are very having a very high power of regeneration, which means if you even if you crush some of these sponges, or even if you crush and make it completely as a homogenate, even then if there are live cells present, they could be able to recover from that particular type of damage, and they could be able to give you the complete cell. The organization of the sponges are grouped into three, which are like ascon type, cycon type, and the leuconoid type. So the sponges can be classified into three different groups. Either it could be of ascon type, the cycon type, and the leuconoid type. These three classifications are actually being based on to the how the cells look like, and how they are actually interacting with the water, how the water is entering into the cell body and how it is exiting. So it is actually due to the simple and a complex forms. So this is a typical structure or typical sponge what is present, and what you see here is actually a body cavity which is called as the spongiocele. So this body cavity is, as I said, there's no body cavity; it's a aphylomate. Then the opening of this body is called as the osculum. So this is the mouth actually, and then below to this body, it actually contains the small tiny pores, and all these pores are called as the ostium. Through the ostium, it actually receives or it actually filters the water what is coming from the outside, and the sponges can actually do the branching as well as the debranching, and the mechanism through which the sponges are actually acquiring the nutrition is that they are actually filtering the water, and along with the water the food particles are also entering into the body through the ostium, and once they enter into the body, they are being actually being digested by the digestive system what is present in the sponge body, and then the excretory systems are actually going to be go out. Sometimes the ostiums or the pores are also being used even for the oxygen transports and all other kinds of things. Even the ostiums are also being used as a mean to secrete the toxic materials. The examples of the porifera is the Tethrina, Sycon, Granatia, Bupilina, and all these kind of different types of sponges. The body of a sponge is vast or the cylinder like, and it is asymmetrical. So it is actually having different types of bodies. So this body is a cylinder like, and it is typically look like as a vase, and where the opening of this vase is called as the osculum. It is being the body surface is perforated by numerous pores, which is called as the ostia, through which the water enter into the body, and one or more large opening, the oscula, by which the water exits. Which means through this ostia, the water enters into the body, and then from the osculum, the water goes out. And in this process, the water, whatever is present in the water, whether it is nutrients and all other kinds of organisms, it is being filtered, and that's how the sponges are recovering, or sponges are receiving the nutrition. The interior space of the body is either hollow or permeated by the numerous canals lined with the choanocytes. The interior space of the sponge body is called as the spongiocele. So this body is called as the spongiocele, but these are and the body is lined by the cell which is called as the conocytes. So these are actually the conocytes which are actually lining the body, and within the conocytes we also have the pores, and these pores are called as the ostia. The characteristics are skeleton consisting of either the fine flexible spongin fibers, siliceous spicules, or the calcareous spicules. So in the different types of sponges, you can have the internal skeleton which is made up of the fine flexible spongin fibers. So it could be of the spongin fibers or the siliceous spicules or the calcareous spicules. There is no mouth present in the in the sponges, and the digestion is intracellular, which means the material whatever comes into the body through the oscula through the ostia is being digested by the digestive enzyme what is present in the spongiosule, and then the toxic materials are going out, or the water is also going out through the osculum. They have a very primitive nervous system. So nervous and the sensory cells are not very differentiated, and they have a very primitive nervous systems where the neurons arrange in a definite network of bipolar or the multipolar cells in some, and because of doubtful status. So there is not a well-defined nervous system present in these cells, and also apart from the animals which actually respond when you are actually giving them some kind of stimulus, uh, the sponges are not very sensitive in terms of, and because they don't respond. So because there is no locomotion present in the sponges, and this is very distinct property of the members what are belonging to the Porifera, and that is the reason behind this is because they don't have a very well developed sensory system. Sponges are the monoecious, which means there is no distinction of the male and female. So they are actually monoecious. The reproduction occurs by the both sexual as well as the asexual methods. The asexual method reproduction occurs by the birds and the gemmules. So what happens is the sponges, as I said, sponges are attached to the rock, and then you have the sponge bodies, and so when the sponge body is branching and debranching, one of the branch actually can get broken down. And for example, suppose this branch is going to be broken down, or suppose this main body is actually being broken down into the multiple pieces. And then what will happen is all these multiple pieces are actually going to float into the water, and then they will go away from their the parent sponges, and then they will go and stick to the different rocks, and when they stick to the different rock, they are actually going to develop the osculum. They are actually going to develop, and they will going to regenerate. So sponge is split or broken into the multiple pieces either by accident or by fission, and then these multiple pieces are again gone away from the parents, and then they will be keep floating into the ocean, and until they don't find the rock or some surface through which they can be able to stick. And once they got stick, then there will be a regeneration of the remaining part. Like if the sculum is absent, then they will develop the osculum and cell body and all that, and that's how the new clones attach to a surface and begin to grow. If you don't know, then actually this is the mechanism through which the sponges are being produced in the ocean. So what people do is they actually are using a rock, and these rocks are actually being tied up with the help of the chains, like the metal chains. And what they do is they just take the small amount of the sponges. Like for example, you take the small amount of sponges and then you inoculate that sponge onto a rock sample. And then what you do is you just put this whole rock into the ocean with the chain, and this chain is being tied up to the corners, right? So you can imagine that I have tied up a big rock with the help of the chain, and that chain is binding to a surface outside, right? So then this rock is actually floating into the ocean on this side like this, and then this sponge is actually going to grow, and it will grow all over the place into this particular rock. And ultimately when they want to harvest, what they'll do is they will take out this particular rock, they will remove the sponges and whatever the sponges are being grown, and that's how they will be able to recover these sponges. Because one of the commercial use of these sponges is that they are being used for different types of sponges what you use in your home. Like for example, I'm not talking about the sponge what you use which are actually of the synthetic, but the sponge, like many people use the sponges for bathing and all other kinds of purposes. And because the sponges are very high power of regeneration. Sexual reproduction occurs by the ova as well as the sperms. So all the sponges are hermaphrodites, which means they are bisexual. So there is no, so male and female both are present. And how they are doing the sexual reproduction. In the sexual reproduction, the one male is one sponge is actually behaving as a male, like so it is behaving like a male, and the other sponge is actually behaving as a female. Although both are hermaphrodite, both are bisexual, but one of the sponge is actually producing the sperm cells, whereas the other kind of sponge is actually producing the egg. So what happen is the male is producing the sperms, and these sperms are, by the help of the water, they are entering into the with the help of the osculum, they are entering into the neighboring female body, or where the sponge is uh, having the egg. So the sponge enters into another sponge. A sperm cell fertilizes with an egg cell. So eggs are present inside and they get fertilized. And then the larva is going to be developed, and the larva is going to come out with the from the opening, and then it will actually go and stick to another rock, and that's how this larva will actually going to develop the sponge. Uh, so the cleavage is holoplastic, which means every time it is actually going to have the clear cleavage. Development, it is direct through a free swimming ciliate called larva, or the amphiblastula or parenchyma. So this larva is also called as the amphiblastula or parenchyma. Sexual reproduction occurs by the ova and sperms. Fertilization is internal. So fertilization is internal when it is when it is occurring inside the cell body of an organism, and the cross pollination always occurs. So it's not as the self pollinations, it is the cross pollination, cross fertilizations, where the sperms are going to come from the neighboring sponge, and it will going to be received by the another sponge. So this is all about the simplest organism, the Porifera, which is belonging to simplest organism, the sponges, which are belonging to the phylum Porifera, and they are actually having the cellular level organizations, asymmetrical and the acoelomates. Now let's go to the slightly higher level of organizations. So next to the cell, you are going to have the tissue level organization. So within the tissue level organization, you can have the radial symmetry or the bilateral symmetries. So within the radial symmetry, you can have an organism which could be acoelomates, which means it does not contain the true coelom. And within this properties or the feature, with these properties and feature, you can have the two phylum. One is called as the Coelenterata, the other one is called as the Ctenophora. So let's start a discussion about the Coelenterata, which is actually a acoelomate, radial symmetry, and it is actually having a tissue level of organizations. So the phylum Coelenterata is colonial or the solitary forms. So they are actually being present as a colony, or they are also free flowing, free living. They are sedentary or the free living animals. So just like the Porifera, they are also either attached to a substratum, or they are free living organisms. Mostly marine, but could be found in freshwater like hydra. So hydra is a very classical example what is present in the Coelenterata, but that is not present in the marine. It is present in the freshwater. The body is readily symmetrical. The tissue level, there is a tissue level organization. There is no organ present. The body cavity, called as coelentrion, helps in digestion and the circulation. So the body cavity, which is thick like this, is called as the coelentron, and this is a typical hydra what I am showing. There are two different types of the cell bodies present in the phylum Coelenterata. Either it could be of polyps or the medusa. So what is mean by the polyps is, the classical example is the sea anemone, that it is actually attached to some surface, like like rocks or the cell bottom or the bottom of the ocean, whereas the medusa are actually the free flowing, and they are like the classical example is the sea pen or the hydra. uh, the polyps are sedentary, so they don't move, right? Whereas, and the cylindrical life forms, the medusas are free living, swimming, and umbrella shapes. The examples are Physalia, sea anemone, sea pen, and the hydra. Sea anemone is a polyp type, whereas jellyfish is a medusa type. So this is a jellyfish where you might have seen the jellyfish, which is having the umbrella-like head, and you have the other body parts. So in the hydra, you have digestion as well as the circulatory system occurring within this cell body, which is called as the cylindron. The mouth of, for example, this is a hydra, right? So mouth is surrounded. So this is the mouth, the top portion, right? And mouth is surrounded by the tentacles, which helps in locomotion as well as capturing the prey. So these are the tentacles which are being used for two purposes. They are actually generating the current, and that's how the hydra actually can move because it is attached to a surface. So it actually can swim in this direction in a swinging motions, and that's how it actually can reach to different types of prey, and that's how they can be able to capture the prey with the help of these tentacles. The tentacles actually have the specialized cells called cnidocytes, which actually contain the stinging structure called as the nematocytes. So on these tentacles, you have are made up of a cell which is called as the cnidocytes, and on these cnidocytes, you also have the stinging structure which are called as the nematocytes, and these nematocytes are very potent. So once the hydra actually catches a prey with the help of these tentacles, the the nematocytes actually discharge the toxins, and that's how the prey is actually going to be either going to be died, or it is actually going to be not going to run away, and that's how it is actually going to then catch the prey, and then the prey will enter into the cell body, and it is going to be digested by the digestive enzyme what is present in this cell cavity, and ultimately the nutrients are going to be used by the hydra, whereas the toxic material are again going to be thrown out. So what you see here is there is no anus present. The mouth is serving as the entry of the food, and the mouth is also using to throw the waste. So mouth is serving as the mouth as well as the anus. It's a poorly there are poorly developed the nervous system. So you can see here is the nervous system where all there are nerve cells what is being connected to each other and forming a nerve networks. Reproduction is both the asexual as well as the sexual matter. So hydra reproduce asexually by producing the birds in the body wall, which grows to be a miniaturized adult and then break away when they are mature. When the hydra is well fed, a new bird can be formed every 2 days, which means you can see here, right? So by there will be a budding into the hydra, right? So a main hydra, the mother hydra, is actually going to have the different types of birds, and all these birds are actually going to develop into an individual adult, and later on this bird is actually going to be detached from the hydra, and then it is actually going to bind to a surface, and that's how it is actually going to start the new hydra. In the sexual means, it involves the gametes. So how the sexual reproduction occurs. So in the sexual reproductions, what you have is you have a male, which is actually bisexual thing. So it is actually going to produce sperms. Sperms are actually going to be received by the another hydra, and that's how it is actually going to have the fertilizations. Post fertilizations, there will be which is actually going to form the embryo, and then this embryo is actually going to develop into the young hydra, and then the young hydra is actually going to bind to a rock or some surface, and that's how it will grow and form the adult hydra. So this is all about the Coelenterata. And now let's move on to the another phylum, which is called as the Ctenophora. So phylum Ctenophora are the exclusive marine animals. So they are not present in the freshwater compared to that the Porifera as well as the Coelenterata. Both are present in the freshwater as well. Then they have the radial symmetry. So you can see here, right? It has a radial symmetry. This ball can be dissected into multiple ways, and that's how they have the radial symmetry. This the Ctenophora animals are diploblastic, which means they are going to have the two germ layers. They are going to have the ectoderm, and they are going to have the endoderms, and they are not going to have the mesoderms. So mesoderm is missing. These animals are so called as the sea walnut or the comb jellies. They have a spherical body with external row of cilia. They exhibit the bioluminescence. So I'm sure you all probably know about the bioluminescence. Bioluminescence means it is actually going to produce the light, and that light is actually not going to be produced by simply absorbing the light from the sun. The light is being produced by the organism, and this phenomena is called as the bioluminescence. Um, the, how this phenomena is occurring is when the organism is actually running some of the metabolic reactions, and because of those metabolic reactions the ATP is being used, and ATP, you know that in triphosphate ATP is being utilized, and that ATP actually is going to be burned down to produce the light. And I'm sure in you might have discussed about the bioluminescence, or you might be going to discuss about the bioluminescence. These are the bisexual animals, so male and females are both present in the same organisms. Reproduction by the sexual reproduction, and the sexual reproduction is occurring at the external fertilization, which means all the ova as well as the sperms are going to be thrown out from the organisms, and then they are going to have the external fertilizations. The examples are Ctenoplana and Pleurobrachia. So with this, we have discussed about the cellular level of organizations. So we discussed about the phylum Porifera, which are the simplest organisms. Then we discussed about the tissue level of organizations, where we have discussed about the radial symmetry, and within the radial symmetry and X-like xylomate, we have the two phylum, which is called as the Coelenterata as well as the Ctenophora. Now let's move on to little more complicated, where we can have the organ or the organ system as the organizations, and within this you can have the bilateral symmetry. And bilateral symmetry is having the very different types of options, like the place where you can have the no body cavity acoelomates, or the pseudocoelomates, or the coelomates. So these are the organisms which have the true cilium. So let's start discussing about the organism which does not contain the body cavity, and the phylum what the organism where the phylum to which it belongs is called as the Platyhelminthes. So Platyhelminthes, the phylum Platyhelminthes is having a bilateral symmetry animal with the blind sac the body plan. It is a triploblastic and the acoelomic animals. They are mostly endoparasites, which means they are going to be feed onto the another host, and they are actually going to be present inside the body. The body is a ribbon-like, so you can see here this is a tinea solium, and it is a flat ribbon-like structure, and that's how the this is also called as the ribbon tapeworm, because this is present just like a tape, right? If you have used some, if you might have seen the tape in your home, with mostly the mothers are using the tape for measuring the cloth and all of the kinds of things. So body is a ribbon-like, unsegmented, and dorso-ventrally flattened and covered with the cuticles. So this body is also having a cuticle. So cuticle is like a kind of a covering. These are the parasitic forms have show the suckers or hook for attachment to the host. So they are actually having a suckers. These suckers are being used to suck the nutrition from the host, whereas it also has the hook which is on the mouth part, and these hooks are being used to actually hook to the body or the wall of that particular animal. So mostly these worms are present in the intestine, so human intestine or the vertebrate intestine. So they actually use these hooks for attaching to the intestine wall, and then with the help of the suckers, they actually sucks the nutrients either, or they also suck the blood. So they can actually damage the wall of the intestine, and then they can directly suck the blood from the human beings, or they can actually take up the digested food from the human. So, and that's why they don't have a very well defined digestive system, because they don't require the digestive system, because what they food they take from the humans are actually the digested food. Excretory system is made up of the phlegm cells, and the classical example is the tinea solium and the fasciola. So tinea solium is called as the tapeworm, and the fasciola is called as the liver fluke. So liver fluke is a very big problem for all the animals like the buffalo, cows, and all that, and it actually causes the cancer, because when it grows into the liver cells actually get transformed, and that's how it actually develops the cancer into the animals. So this is what a cross section. What you see here is a stomach, right? This is the stomach of the tinea solium, and this is the sensory papillae. And so as and then you have the nerve cords, which actually grows and runs from the central portion to the end of the things, and then by the end of this cell body, you have the anus. So all the things are going to be taken up by the tinea solium, and then the waste material is going to be ejected from the anus. The nerve system is in the form of a nerve ring or the nerve cords. So this is the nervous system. You have the central ganglia in the front, and then you have the nerve cords, which is running from the top to bottom of the cell body, and then you have the appendages coming from the nerve rings, and so that they can be able to sense the uh, the organism belonging to the phylum Platyhelminthes are hermaphrodites, which means they are actually going to have the bisexual. Uh, so reproduction is well developed. The male and the female sex organs are present on the same body. It has a great power of regeneration, so it can be able to regenerate. So this is a reproductive system where you can have the female reproductive organs, ovary, or the female or the male reproductive organs like the testes, which is present onto the same cell body. So you can imagine that this is one of the segment which is present in the or one of the cross section of a segment within the this tapeworm, where you have the testes. So these are the testes actually, which are actually going to produce the sperm, and these are the ovaries. So they will produce the egg, and that's how there will be internal fertilizations. The locomotory structure and the sense organs are absent, because there is no locomotion required in these particular organisms, because they will, once they reach to the intestine, and they will actually going to stuck to the intestine with the help of the hooks, and then they will actually going to start taking up the nutritions. So this is all about the platyhelminthes. Now let's move on to the slightly more advanced organisms, and the least advanced organisms are having the pseudocoelomates. So they don't have the true coelom, and the example is Aschelminthes or the roundworms. So phylum Aschelminthes, they are commonly called as the roundworms or the worms. They are mostly parasitic, living in the body fluid of the host. The body is long and cylindrical, which is thread-like. So this is a body they are not as big as the tinea solium, and they are not as flat with the tinea solium. So that's why they are round. Actually, they are round worms. These are the the classical worms what is present in the stomach of or most of the children's, and they are the bilateral symmetrical animals. So they have if you cut it into the center, they are actually going to be divided into two halves. They are triploplastic with the perivascular cavity. They are pseudocoelomates. So there is a cavity what is present in the Aschelminthes organisms, but that is a pseudocoelomate. It is get filled with the material. These animals show the tubes within a tube-like body plan. What is mean by the tube within the tube-like body plan? So you can have a body plan like two ways. One, what we have discussed just before, like for Porifera or Coelenterata, where on the one side you have a mouth, and from the mouth the things are entering into a body, and then they are being processed. Either they are going to be digested and all that, and then the digested product is coming out. So this is called as the blind sac plan, where there's no anus actually, and there's no alternate route. So the material will enter into the body, and then they will pass through, processed, and then it will go out. Whereas in the subsequent matter, what you have is you have a cell body from outside, and within the cell body you are going to have a mouth, and mouth is connected to another tube. That another tube is actually a alimentary canal, and alimentary canal is going to have the two openings, one for the mouth, the other one is for the anus. And this kind of arrangement is called as the tube within the tube, which means you have a tube which is actually the external body, and then you are going to have a tube which is inside. So that's why this kind of arrangement is called as the tube within the tube body plan. Whereas in this particular type, you have a blind sac, which means you are only going to have the external body, you are not going to have the internal body, internal tubes, and where you are going to have a mouth in the front, and that mouth is actually going to receive the material, and that mouth is also going to, it's going to receive the material and also going to be used for throwing out the toxic material. The body walls shows the longitudinal muscles, but no circular muscles. Excretion takes place by the protonephridia. The roundworms lives on average of 4 months and have a life cycle ranging from as early as 14 days to as long as the 80 days, depending on the species. So they have a average lifespan of the 4 months, where some of the members may have the lifespan of 14 days, whereas some of the members may have a lifespan of 80 days. The classical examples are Ascaris, Vaucheria, and Dracunculus. Uh, Phylum Aschelminthes, nerve ring have a nerve ring or nerve cords. The reproductive system is well developed. It is unisexual, which means it is going to have the separate male and females. So this is the male, this is the female. What is present in the Ascaris? So there is a sexual dimorphism. What is mean by the sexual dimorphism is that there will be a distinct characteristic of the male as well as the female. Sexual dimorphism is also present in many other organisms, like the humans also. Like you might have seen the male is very different from the female. Similarly, in the Aschelminthes also you have the sexual dimorphism. So male Ascaris is shorter than the female, so that is one characteristic, right? The male is very short compared to female, and the male Ascaris has a curved posterior end with a penile situ for the reproduction, which means the male is short, and the male is also having a posterior portion which is curved actually, and it also contains the penile situ for the reproductions. Whereas the female Ascaris is long and it is relatively straight. So you can see that this is very big and it is actually a straight. Fertilization is internal or maybe cross or self. Development may be direct with or without any intermediate port, or the indirect. Various lateral lines and ports are present on the surface of the body. So how the Ascaris is actually infecting a healthy human being is that when you are taking a food or you are drinking the water which is contaminated with the Ascaris egg from the feces, so what happens is when somebody is getting Ascaris infection, it is actually doing the when it is so it is present in eggs are present in the soil, and when you are taking the food which is not properly cleaned, or when you're taking a contaminated water, these eggs are present in that, and once you take them through the mouth, they will go into the intestine, like they will go to the small intestine, and from the small intestine they won't stay in the small intestine for very long. From the small intestine, they make a hole into the intestine, and that's how they reach to the lungs. They migrate to the lung, and from there, once they enter into the lungs, they will actually going to come out with by the coughing, right? Or they can be cuffed, and then they will enter into the mouth, right? And then they will again going to be swallowed. Once they got swallowed the next time, by the time the ascaris is being fully developed, and now it will reach to the intestine, and it is actually going to stuck to the intestine, and it will start replicating. The eggs hatched in the intestine burrow through the gut wall and migrate to the lungs via the blood, right? So it will make a hole into the gut, they will, and it will reach to the lungs. In the lungs, actually, they will going to do all the development. So they will going to reach to the alveoli, they will pass through the trachea, and where they are actually being going to come out by the coughing, and then once they come out from the coughing, they will reach to your mouth, and then they will get swallowed. They develop into the adult worm in the small intestine, and the adult worms generally eat the food of their host, but heavy infection can cause the tangling of the wounds which can obstruct the guts. See, what happens is that when it is present in the small intestine, first thing is in the small intestine, it is actually going to get the completely digested food, and if there will be a small number of ascaris, then it is fine. If it is a large number of ascaris, then what will happen is that the small intestine is connected to a large intestine, and when the number will go up, it is actually going to obstruct this particular type of connection, which is connecting the small intestine to the large intestine. And at that point, it is actually going to cause the severe stomach pain, and that time there will be no other option but to go for the surgery, and then you have to remove these worms from the small intestine. That's why there is a recommendation that you take the anti-helminthic and as well as the anti-worm tablets on every 6 months, so that they will give, if there will be a injection of the eggs by any mean into your system, all these anti-worm medicines like albendazole or mebendazole, all these are actually going to kill those eggs as well as the adult as well. So with this, we have discussed about the cellular level of organizations. We discussed about the phylum Porifera, and we discussed how the Porifera is acylomate and mostly asymmetrical organisms. Then we moved on to a slightly more advanced stage where we have discussed about the tissue as well as the tissue system. And within the tissue, we have discussed about radial symmetry, acylomate, and we discussed about the Coelenterata as well as the Ctenophora. And then we discuss about the organ as well as the organ system of organizations. Within that, we discuss about the bilateral symmetry, and we discuss about the organism which does not contain the verticavity like acyllomates. The classical example, we discuss about the Platyhelminthes. So we discuss about the Tinea solium and how the Platyhelminthes are under the proper and the characteristics of Platyhelminthes. And then we discuss about the pseudophylomates, and within the pseudophylomates, we also discuss about the Aschelminthes. So with this, I would like to conclude my lecture here. In our subsequent lecture, we are actually going to discuss about the organism which are having the organ or the organ system, bilateral symmetry, and the true coeloms like the coelomates. And within that, we are going to discuss about the Annelida, Arthropoda, Mollusca, Epidermidata, Hemichordata, and Chordata. So with this, I would like to conclude my lecture here. In the subsequent lecture, we discuss about the coelomates. Thank you.